Good day, grade 11 learners, and welcome to this video where we will be revising the second topic which will be tested in your Life Sciences Paper 1, namely cellular respiration. The following are important concepts which you will need to remember when studying the topic on cellular respiration. Firstly, you need to know the process of aerobic respiration. The process of aerobic respiration involves glycolysis, the Krebs cycle, and oxidative phosphorylation. Make sure you know exactly the steps that are involved in glycolysis, the Krebs cycle, as well as oxidative phosphorylation. The second type of cellular respiration, in addition to aerobic respiration, is anaerobic respiration. That is when living organisms are going to undergo cellular respiration in the absence of oxygen. So the first type of anaerobic respiration occurs in plants. That was alcoholic fermentation. Secondly, in animals, the type of anaerobic respiration will be lactic acid fermentation. You need to know exactly what the products are of alcoholic fermentation as well as lactic acid fermentation. For example, alcoholic fermentation produces ethanol, carbon dioxide, and a small amount of energy, but lactic acid fermentation produces lactic acid and a small amount of energy. Then you need to know the differences between aerobic as well as anaerobic respiration. You will normally need to tabulate those differences. You need to know how to draw the mitochondria, the drawing of the mitochondria, as this is the structure in which the process of cellular respiration takes place. The organelle in which cellular respiration takes place, the mitochondria. You need to be able to draw and label that structure. Lastly, we looked at practical investigations. The type of practical investigation that was focused on was the one where we try to determine if carbon dioxide is released in the process of cellular respiration. The first question taken from our exam question papers, we will look at question 3.5. The instruction says read the passage below. Both aerobic and anaerobic respiration are used to supply energy during exercise. During some types of exercise, such as sprinting, the body is unable to supply enough oxygen to the muscles and lactic acid builds up in the cells. This is because the body cannot inhale enough oxygen to meet the muscles' requirements, and the muscles go into oxygen debt. This debt can be repaid breathing when the sprint ends. In a 100-meter sprint, the total oxygen required by the muscles may be as much as 8 liters per minute. It is impossible for the body to deliver this amount of oxygen during the few seconds it takes to run the race. The maximum oxygen that can be taken into the body is about 4 liters per minute. The first question asks you to write down a word equation for aerobic respiration. For 2 marks, you are writing down a word equation for aerobic respiration. So remember that cellular respiration, specifically aerobic respiration, has the equation glucose plus oxygen is going to release carbon dioxide, water, and energy, which is going to be in the form of ATP. 3.5.2 asks us to describe how lactic acid is formed in the muscle cells. For 3 marks, we're describing how lactic acid is formed in the muscle cells for 3 marks. Now, if we go to the paragraph, we can see it says during some types of exercise, such as in sprinting, the body is unable to supply enough oxygen to the muscles, and the lactic acid builds up in the cells. They are asking us to describe how lactic acid is formed. The paragraph does not give you enough information to describe how that lactic acid is formed. Therefore, you need to answer the question based on your prior knowledge. How is lactic acid formed? You need to first think about lactic acid fermentation. What happens in lactic acid fermentation? Lactic acid fermentation takes place in the absence of oxygen. Glucose is broken down into pyruvic acid, and then pyruvic acid is going to form lactic acid. So if we look at the answer to 3.5.2, glucose is broken down, the process of glycolysis occurs. It occurs in the absence of oxygen, and the glucose is broken down into two molecules of pyruvic acid. The pyruvic acid is then converted into lactic acid. So the question is for 3 marks. Any 3 of these points would have given you 3 marks for the question. 3.5.3 says explain how the oxygen debt referred to in the passage is repaid for 3 marks. So this oxygen debt, it states here, this debt can be repaid breathing when the sprint ends. So this debt can be paid breathing when the sprint ends. The question is asking you to explain how the oxygen debt referred to in the passage is repaid. So how does this oxygen that you did not have, how did it eventually come back? So firstly, after exercise, the rate and depth of breathing is going to start increasing. After you've engaged in that strenuous exercise activity, your rate and depth of breathing starts to increase. That is part of your understanding with regards to homeostatic control of breathing. The rate and depth of breathing will increase. When your rate and depth of breathing does increase, you're going to have more oxygen transported to your muscles. The lactic acid will then be converted back into pyruvic acid. So the lactic acid which you initially had at the start gets converted back into pyruvic acid, and this will now allow the normal aerobic respiration to occur. So I'm going to go through that again. After the strenuous exercise activity, the rate and depth of breathing increased. You now have more oxygen being transported to your muscles. The lactic acid which you initially had will be converted into pyruvic acid. And once converted into pyruvic acid, the normal aerobic respiration can now occur. So you can now move on to the Krebs cycle and oxidative phosphorylation. 3 marks for that question. 3.5.4 states that the runner took 12 seconds to complete the 100-meter sprint. The runner took 12 seconds to complete the 100-meter sprint. Calculate the amount of oxygen in liters that you need to inhale per second to prevent going into an oxygen debt. For 2 marks, we are calculating the amount of oxygen in liters. The runner took 12 seconds to complete the 100-meter sprint. Let's go back to the paragraph and see if there's some useful information that could be of assistance to us. So it says in a 100-meter sprint, the total oxygen required by the muscles may be as much as 8 liters per minute. There we go. We could possibly use this. 8 liters per minute. And the runner took 12 seconds to complete the 100-meter sprint. You want to calculate the amount of oxygen in liters. There you have oxygen in liters. And what is your value that you have? 12 seconds. You can convert that 12 seconds into minutes and then multiply by 8 to get the number of liters. Therefore, for the answer, you will take your 12 seconds and you divide by 60 because there are 60 seconds in 1 minute. You multiply by 8 and therefore you get 1.6 liters. 12 seconds divided by 60 seconds multiplied by 8 liters gives you 1.6 liters. 2 marks for that question. The next question for 3.2 states in cellular respiration, glucose is broken down in a step-by-step process to release energy as ATP. Glucose is broken down in a step-by-step process to release ATP. So firstly, in 3.2.1, we are describing the events that occur from the moment glucose is broken down until carbon dioxide is released for 7 marks. What are we actually looking at here? We're describing the events of cellular respiration from the moment glucose is broken down until carbon dioxide is released. So you first have to ask yourself the following question. In which stage is glucose broken down? It's broken down in glycolysis. Next question you ask yourself, in which stage is carbon dioxide released? It's released in the Krebs cycle. So when you are going about answering your question for 7 marks, what will your answer comprise of? It will comprise of a description of glycolysis, and it will also comprise of a description of the Krebs cycle. Therefore, the answer will be as follows. Firstly, you need to state glycolysis. You're going to get a mark for stating glycolysis, and the word glycolysis must be there. If the word glycolysis is not there, you will be penalized. So what is glycolysis? Firstly, it occurs in the cytoplasm. Glucose is broken down to form 2 molecules of pyruvic acid. The molecules of pyruvic acid, after it's broken down, you're going to release energized hydrogen atoms. Some of the energy are released to form 2 ATP molecules. That is when it's carried into the third process, which is oxidative phosphorylation. The next phase will be obviously the process is controlled by enzymes. The next phase, the Krebs cycle, you need to write down the word Krebs cycle. There's a star next to it, which means that if the word Krebs cycle is not there, you will be penalized. 1 mark for this question. So the Krebs cycle, pyruvic acid enters the mitochondria. A series of cyclic reactions occur. Energized hydrogen atoms are released, which is taken to oxidative phosphorylation. That's the third phase. And that's the end of it. The next question looks at 3.2.2, where you are drawing a label diagram that shows the organelle in which cellular respiration occurred. A label diagram of the organelle in which cellular respiration occurs, 5 marks. The organelle in which cellular respiration occurs is the mitochondria. Therefore, we draw the mitochondria for 3.2.2. This is the drawing of the mitochondria. You have a heading at the bottom that says drawing of the mitochondria. You will need to write drawing of the mitochondria. You have at least 3 correct labels: the outer membrane, the ribosome, the cristae, the inner membrane, the mitochondrial matrix, and the DNA. This is the criteria that will get used to mark a drawing like this. 1 mark for your heading. 1 mark if you have the correct drawing. If you drew a mitochondria, 1 mark. If you drew a chloroplast, you lose that mark. It's the incorrect drawing. Any 2 correct labels in this case, you will get a mark. Because this question is out of 5 marks, you would have had an additional mark awarded here for you. Instead of 2 correct labels, you would have had 3 correct labels. So that you have 3 plus 1 gives you that total out of 5. So for this question, that's 5 marks. You would need 3 correct labels. If the mark allocation was out of 4, you would have simply needed 2 correct labels. 3.2.3 states that you need to state the differences between the 2 types of anaerobic respiration in terms of the products they release for 3 marks. You are not tabulating. You are simply stating the differences between the 2 types of anaerobic respiration in terms of the products they release. There are 2 types of anaerobic respiration. It's lactic acid fermentation and it's alcoholic fermentation. So in lactic acid fermentation, what are the products? Lactic acid and a small amount of energy. In alcoholic fermentation, what are the products? Carbon dioxide, small amount of energy, as well as ethanol is also a product of alcoholic fermentation. So for 3.2.3, lactic acid fermentation releases lactic acid, and a small amount of energy. Alcoholic fermentation releases ethanol as well as carbon dioxide and a small amount of energy. Apologies for this question number. Question 2.4.3 should have stated 3.2.3. So this should have said 3.2.3. That is the answer to 3.2.3. The next question that we'll focus on is an experiment question. So 3.1, an experiment was performed in a laboratory to investigate the effect of the concentration of sucrose on the rate of cellular respiration in yeast cells. To investigate the effect of the concentration of sucrose on the rate of cellular respiration in yeast cells, that is definitely our aim of the investigation. The following steps were followed. 4 test tubes were used, each with a different concentration of sucrose. Test tube A had 100 milliliters of water and no sucrose was added. Test tube B had 0.5 grams of sucrose result in 100 milliliter of water. Test tube C had 1 gram sucrose in 100 milliliter of water. And test tube D had 1.5 grams sucrose in 100 milliliter of water. The same species of yeast cells was added at each test tube. A small amount of salt was added to each test tube to slow down the rate of cellular respiration. Each apparatus was allowed to stand for 10 minutes. The volume of gas produced by the yeast cells in each test tube was measured and the results were tabulated as shown below. Firstly, 3.1.1 asks you to identify the dependent variable. Remember, the dependent and independent variable we take from the aim of the investigation, which we highlighted at the start. So to investigate the effect of the concentration of sucrose on the rate of cellular respiration, the dependent variable is the one you are measuring. What are you measuring in the statement? The rate of cellular respiration in yeast cells. Therefore, the answer is the rate of cellular respiration. Next question, you are identifying the independent variable. Which variable are you changing here? The concentration of sucrose. So the sucrose concentration is the answer. 3.1.2, you are naming the gas produced by the yeast cells for 1 mark. What gas should be produced by the yeast cells in this process? If we are testing for cellular respiration, what gas is produced? Carbon dioxide is produced. If we look at the following table, it's showing us the results of the investigation. We have our time period is increasing until 10 minutes. The first column is for 0 grams of sucrose per 100 milliliters. It remains to the volume of gas by yeast cells in milliliters. It remains 2 milliliters. When 0.5 grams of sucrose was added, it increased. When 1 gram of sucrose was added, it increased as well, even further. 1.5 grams of sucrose was added, it increased even more. It went up to 9.4 milliliters. 3.1.3 asks you to explain how the investigators could test to verify the gas named in question 3.1.2. And what was that gas? That gas was carbon dioxide. So for 2 marks, how are we going to verify that carbon dioxide was released? You would have remembered one of the experiments that we covered in cellular respiration was to determine if carbon dioxide is released as a product. In order to accomplish that, we used clear lime water. The clear lime water needed to turn milky. That can be used as an indication. If clear lime water turns milky, it will prove that the gas carbon dioxide is actually released. 3.1.4, we are naming 2 ways. You are naming 2 ways in which the investigators could have increased the validity of this investigation. How could the investigators have increased the validity? Firstly, They could have made sure that all 4 test tubes were at the same temperature. Secondly, they could make sure all 4 test tubes have the same oxygen concentration. Why the same oxygen concentration? Because if the oxygen concentration changes, it could have had an effect on the results. Thirdly, you add the same amount of yeast to each test tube, because the investigation is on yeast here. You add the same amount of salt to each test tube, and that was mentioned as well. So 2 ways in which the investigators could have increased validity. Any 2 of these would have given you 2 marks. The next question, 3.1.5, says explain the significance of the inclusion of test tube A in the experiment. Why are you including test tube A? So you would have remembered in test tube A, this was the one with 0 concentration of sucrose. And you notice the volume of gas remained the same. So why are you going to include a test tube like that? Because that test tube will serve as a control. And it will ensure that the results obtained are due to changing sucrose concentration. So it's going to serve as a control to ensure that the results are going to be valid. And it's also going to allow you to have something to compare your results of the investigation with. Next question, 3.1.6 asks you what conclusion can be made based on the information in the table for 2 marks. So based on this information in the table, what conclusion can you make? As you can see, you are increasing your sucrose concentration. And when you increase your sucrose concentration, what else is increasing? The volume of gas produced by yeast cells. Therefore, the answer for 3.1.6, the higher the sucrose concentration, the higher the rate of cellular respiration. Or you could also say, as the sucrose concentration increases, the volume of gas produced by yeast cells increases. And therefore, it increases the rate of cellular respiration. 3.1.7, you are explaining how this process in yeast cells can be used to produce alcoholic beverages like beer for 3 marks. What are they talking about here? Explain how this process in yeast cells is what? Plant cells. Yeast cells are found in plants. Can be used to produce alcoholic beverages. Alcoholic beverages, what does this mean? Which type of fermentation are we looking at? Alcoholic fermentation. So what are we explaining? We are explaining alcoholic fermentation. 3.1.7, firstly, in the absence of oxygen. Why? Because anaerobic respiration takes place here. In the absence of oxygen, yeast cells will undergo alcoholic fermentation. What happens in alcoholic fermentation? You are going to be producing alcohol or ethanol as well as carbon dioxide. That's the products that you will yield. 3 points for 3 marks for that question. That brings us to the end of the section where we revised cellular respiration. It was the second topic which you will need to study for paper 1. We already completed photosynthesis. We have now completed cellular respiration. Please make sure that you are studying the correct topics for paper 1 of your final exam. Thank you so much for taking the time to watch this video. The next video will be focusing on animal nutrition as the third topic that you will study for paper 1.